after the ovulations, as you know, when the follicles, the Graafian follicle is ruptured, the oocytes with some granular cells that name is coronal radiata coming into the uterine tube in the same time of the ovulation as a result of contraction of the fimbriate part of the uterine tube. The oocytes is coming to the uterine tube. It's the oocyte can alive around 6 to 24 hours in the uterine tube, and if the sperm is coming, we have a fertilizations. Before the fertilization, as you know, the sperm cannot fertilize the oocytes. The sperm have to doing a two process for fertilization of the oocytes. One of them is name is capacitation. Another one is name is acrosomal reaction. What this means? The first of all, the capacitation during the passing sperm from the epididymis and epididymis in testes, there's some excess protein added to the surface of the sperm, spatially membrane of sperm over the head regions. In the time of capacitation that take place in the female genital tract is take around 7 hours, and during this period some excess protein is removed from the surface of the sperm membrane, and this excess cholesterol again is removed from the sperm membrane, because this cholesterol causes the rigidity in the membrane. As a result of the uptake and removing of this cholesterol, the sperm membrane be fluid and flexible and is ready for the fertilization. Another things that's happened during the capacitation are the increase of the calcium ions, increase of pH, and increase of the bicarbonate in the sperm cytoplasm. As a result of increasing of the calcium, the sperm be hyperactivated, and this hyperactivated is important for the movement of sperm toward the oocytes. When the oocyte receive in the ampullary regions, the fertilization is happen. What this means from the fertilization? Fertilization means fusion of sperm and oocytes that usually it's happen in the ampullary region of the uterine tubes. The fertilization divided to 3 phases. The first phase, you can see these pictures, is passing the sperm through the coronal radiata. For the past the coronal radiata, we don't need any enzyme, because we have a gap between the coronal radiata cells and the sperm. This time is hyperactivated. Movement of sperm help to passing the sperm through the coronal radiata cells. The second phase is happen when the receptor on the surface of the sperm membrane bind to ZP layer, spatially ZP3. As you know, the ZP3 is a kind of the glycoprotein and is specific for the each species. As a result of this binding, we have a acrosomal reaction. During this process, some enzyme that storage in the acrosomal cap are released. The most important enzyme that released during this process, his name is acrosin, trypsin-like enzyme and hyaluronidase. When the sperm passes through the zona pellucida layer, is coming to the pre-uterine space in here, and this time the head of sperm destroy and some receptor in equatorial region, this area, we say that equatorial region of the sperm membrane, we have another receptor like a isomo is an important receptor that's situated in this area of the sperm membrane, and this receptor bind to another receptor on the surface of the ovulema, and this time the third phase is started. When the receptor on the surface of sperm bind to receptor on the surface of the oocyte, the most important receptor on the surface of the ovulema, his name is CD9. These receptors join together, and as a result fusion of oocyte and sperm is happen, and sperm enter to the oocyte and accept the membrane of sperm. The whole of sperm is coming into the oocyte. When the sperm is coming to the oocyte, the first step is happen. His name is cortical reaction. What does means? In the time of the maturation oocytes, some granules is storage and accommodated in the cortical area of the oocyte. We say that cortical granules. These cortical granules contain many enzymes. When the sperm fertilize the oocytes, as a result of exocytosis, these granules release, and when these granules coming out, these granules contain the many enzymes, and these enzymes change the properties of the ZP. As a result of this reaction, we don't have the polyspermia. It means that cortical reaction causes prevention of the polyspermia. The second reaction of the fertilization is oocyte activation, because we need to this oocyte activation for resumption of this the meiosis. As I told you, when the sperm enter the oocytes, bring some activating factor that name is phospholipase zeta. As a result, these activations, another important event that's happened is the resumption of the second meiotic division. The oocyte finishes its second meiotic division immediately after entry of the spermatozoa, and the second polar body released to the pre-uterine spaces. And this metabolic activation is important for the continue of the fertilization. In this picture, in the A, you can see that the finishes of the second meiotic divisions, and when the second polar body you can see in the pre-uterine space, and after the finishing of the second meiosis, around the female genome and male genomes, two temporary membrane are formed, and two pronuclei is formed. This pronucleus, male and female, is essential for the DNA replications. After the replication of DNA in the female and male pronuclei, the first mitotic division it's happened, and finally two cells that name is blastomer are formed. This is the first division, and this is a zygote. When the zygote is formed, the blastomer continue to more division, and this division is named is cleavage. This is a real picture from the human oocytes, the two pronucleus, male and female, and the two blastomer. And this division is named is a cleavage, the 2-cell stage, 4-cell stage, and morula. You can see in each times the size of the blastomer is decrease, and the number of the size of blastomer decrease, and the number of the blastomer increase. And when the blastomer number is received to the around the 16 blastomer, we say that the morula. You can see that the zona pellucida is remain, because if it's dissolved, we have a ectopic implantation, and embryo can implantate it in the uterine tube. It have to remain until to the uterine cavity, and when the zygote received to the uterine cavity, gradually this layer is dissolved and removed, and the embryo is ready for the implantations. When the embryo is coming to the uterine cavity around the 4 days of the fertilization, gradually some fluids is accumulated in between the molar cells, and the embryo cell divided to two parts. The some cells around the embryo is named is trophoblast or outer cell mass, and that cell situated inside is named is inner cell mass or embryoblast. Whole of the embryo tissue are made by the embryoblast cells. When the blastocyst, now we say that this is a blastocyst, and this cavity is named is blastocell. When the blastocyst is formed, gradually the ZP layer dissolves as a result of secretion and fluids in the uterine cavity, and now the blastocyst is ready for implantation. In the end of the first week, some receptors are appear on the surface of the blastocyst, especially on the surface of the trophoblast. And the first of all, the L-selectin is appear, and this L-selectin is necessary for the landing of the blastocyst on the surface of endometrium. And after the landing, another receptor on the surface of the trophoblast, that name is integrin, is important. Integrin is a large family, has many subunits, but that integrin is important for the implantation. His name is alpha 5 beta 3 is important integrin for the implantation. Now some receptor on the surface of the trophoblast and some receptor on the surface of endometrium join together like a key and lock, and blastocyst started to invasion and penetration of the endometrium and coming into the endometrial layer, and implantation is happened. In this picture, for the review, you can see that the harmony between the oogenesis and the uterine cavity growing during the menstrual cycle. Here you can see that the menstrual cycle, as you know, the menstrual cycle takes place around the 28 days from the first day to the 4 or 5 days. We have a menstrual phase. During the 4 to the 14 we have a follicular or proliferative phase. You can see at these times that we have a proliferation in the endometrium, we have a growing in the follicle, and around the 14 days or middle of this cycle, you can see that the ovulation as a result of the LH surge, the remaining part of the follicles making a corpus luteum, and this is the time of the progesterone or secretory phase. You can see that the growing of the uterine glands, and if the fertilization is happen, the embryo is implanted in functional layer of the endometrium, and the corpus luteum be a corpus luteum of pregnancy. Again, the relation between the oogenesis, uterine cycles, and the hormone that's secreted by the adenohypophyse, that FSH and LH, the role of the FSH and LH on the menstrual cycles. This is a first week of gestation. In the next session, I want to talk about the more implantation and the second week of the gestation.